patient who got a transplant for PFIC had the annual anniversary like a couple months ago, and then they brought me this. This is their local soccer team had a championship. So it's a network. It's a network of a worldwide network of PFIC. Okay, let me start it. So this click. So future treatment for PFIC. So this is the first slide. So your task is not to foresee the future, but to enable it. And I need to ask French speaker. I think she stepped out. Okay. So it's from San Digiferi. I don't know how to pronounce so well, but I have a link to this at the end of the slide. So just quickly, let me introduce my research lab. So I'm a half doctor, I'm a half researcher. Our subject is the hepatocyte. We are trying to understand the hepatocyte development, the biology. Our targets are genetic liver disorders, and goals are providing accurate diagnosis and establish novel therapy for genetic liver disorders. Caris, Aitaro, Hieronimo, and Quinn, and Andre is my lab. So I have this disclosure, and thank you for this some sponsorship. And so I have to say, my talk is more, it's a future of treatment. So my talk is very opinionated and biased. So this is my disclosure, but I believe those treatment will be effective. That's why I'm doing the research. So my talk is biased naturally. So overview talk, mapping out the future treatment. What's next? Type of treatment and different approaches. Is it for me? Is it for you? Risk of new treatment. And this is, I use only the example for gene therapy. This is a bridging to the next roundtable talk. So that's why I picked this, and that's why I'm going through this in order. The first topic, this is a big part of the talk. So drug development globally, start with the lab and animal model, and this is preclinical, and then patient trial. This is the clinical trial, phase 1 to 3. And then reviewing by the FDA, and then approval. And then after that, also observation start. So basically at the research development part, it's where the patents get submitted and granted. And at the end, the goal is to get the FDA approval. Today's talk, my talk, I'm focusing on this field, this space. And I pick up these 4 different new emerging therapies: gene therapies, messenger RNA therapy, small molecules, and nutrient bile acid therapy. And I'm omitting a lot of other possible therapies for the time, so for the sake of the time, especially the siRNA, Stacy just talked in the morning. So this is a very global picture. On the top, it's smaller, and then on the bottom, it's bigger because there's many more other types. And then on top, it's less type of gene therapies. And on the top, it's more direct action against the disease and more specific for the PFIC. And then to the bottom, it gets more general and cholestasis alone, not specific for the PFIC. And this is just a perspective. So the bottom ones are basically now, and then top ones are years to come. So this is the perspective. And then, so we start from the bottom one. So nutrient therapy, this is various nutrient, natural substance we just talked about, Somali, actually a lot of a patent already submitted and approved. So, if you search Google patent and PFIC, actually it's a lot of natural substance are already patented. So there is a lot of possible new substance there. One example I want to say it's more different than those natural substances is this choline. Dr. Hayashi submitted and accepted this new preclinical model for PFIC1 deficiency post-liver transplant sheet modeling, and he found IV version or choline supplementation improve the phenotype of the liver steatosis after transplant. So he has patent on this, and he has preclinical and the clinical study in the planning phase. So more to come. and bile acid metabolism modification. This is it's going on. So the clinical trials for other cholestatic diseases are already happening, and some are already in PFIC world too. So reducing bile acid pool is IBAT inhibitors, and there's some more. Reducing bile acid production by FXR agonists, FGF19 analogs, PPAR agonists, and there is new version of RUSO, which is the norcholic acid. It's basically, I would say here, bile acid thinner. There's a much more different function of this, but those are bile acid modifying factors. It's coming to PFIC world. And next one is this small molecule. So small molecules, it works in the hepatocyte. Most of the types of PFIC can get treated with this, but it's only for some missense variants. I will discuss about this later, but let me just stop here. And one example is the chaperone molecule, which similar to Trikafta, which was approved and be effective for cystic fibrosis, especially for BSEP is similar molecule to the molecule missing in cystic fibrosis. So this approach is actively investigated, and it's coming for the clinical trial. And other example for this chaperone molecule is this 4-phenylbutyrate. This was a clinical trial, but it's in the US, it's stopped. But I heard Dr. Hayashi in Japan is still ongoing for this clinical trial, specifically for PFIC patients. And quick biology for families and patients. So we're talking about biology. The basic biology is starting with the DNA. DNA converted to messenger RNA. The messenger RNA produced or translated to amino acid, and this chain of amino acid became a protein. For example, the BSEP has a coding of DNA and become messenger RNA and become a BSEP protein. So this is a gene name is a BSEP and the protein. So BSEP protein, and it get transported to the surface. So this is outside of the cells, this is inside a cell, and then they do the export of the bile acid. So when this DNA become messenger RNA and become a protein, this protein needs to get trafficked to the surface of a cell for there to do a job. So there is a process, even after protein is made, the protein needs to get navigated to the place they have to function. So this small molecule, so coming back to this small molecule, they have an effect called chaperone effect. So those small molecules, some of it has the effect to chaperone this protein to the surface of the cell, so that the protein can function. So in the missense mutation, I'm gonna explain a little more. The protein cannot get trafficked, but if with the help of those chaperone, the protein with the missense mutation can go to the surface and they can work. So this is the mechanism of the small molecule. There is a different other function of small molecules, but this is the one example for you to remember. Okay, so going to the messenger RNA therapy. So the messenger RNA therapy is very simple. You have this messenger RNA inside of this particle running in the blood, and the hepatocyte get uptake and they release this messenger RNA in the cell, and this messenger RNA get converted to protein, and this protein is the one want to have a work and function. So basically messenger RNA is we injecting messenger RNA into the vein, and this messenger RNA is wrapped in the lipid nanoparticle, and the liver is natural organ to accumulate the lipid. So it's a natural destination, not selective, but the naturally liver uptake those lipid, and it's accumulate in the liver. So the liver is very favorable target organ for this approach. And messenger RNA get delivered, but with this messenger RNA has the code of the missing protein. So if you have a BSEP deficiency, we will design BSEP of this messenger RNA. So we can deliver the messenger RNA of a BSEP into the hepatocyte so that they can make it. So, but the caveat is that it's not so selective. So other type of cells, other part of the organ can also uptake. So sometimes it can go to heart, sometimes it can go to liver, kidney, but maybe sometimes the kidney may have a better function there. So, but I have to say this is not specific. And it's similar, very similar mechanism to the COVID-19 vaccine because it was the messenger RNA of COVID, part of the messenger RNA, and in this, I assume in this room, most of the people got this vaccine. So everyone already seen the messenger RNA treatment in your body, tested. So, but it's a downside is the transient nature because the messenger RNA doesn't stay in your body forever. So this approach, you have to repeat injection certain times. We were talking about every 2 weeks or every 3 months or every year. We do not know. Okay, so talking about gene therapy. So Dr. Grumpy talked about gene therapy. So I'm just using very, very brief version and trying to be simple. So basically, gene therapy inject the virus into veins to deliver DNA into cell nuclei, make the cell produce missing proteins. So that's the goal. So this is the difference between the gene therapy versus messenger RNA therapy. So in gene therapy, you put the virus, you put the DNA around the virus capsule, and the virus attached to the target cell, which is the hepatocyte, and then they release DNA. The DNA needs to go to the nuclei, and then in the nuclei, DNA converted to the messenger RNA, and this messenger RNA produce protein. I'll explain for this component, but so this is the gap between how gene therapy DNA go through versus the messenger RNA therapy. Messenger RNA does not go to the nuclei, and they become a protein. Okay, so there's two globally, two versions of gene therapy. One is a transient version, which is genes introduced to nuclei, but it's only transient, meaning the brief time, and it's episomal status. So the episomal means very certain shape of DNA in the nuclei, staying for a brief time. And as time goes, this episomal DNA goes away, so they will lose this therapy effect. There is integrated version. The example is the CRISPR version. This is the famous Nobel awarded technology recently. It's the editing version of gene therapy. And also using the lentivirus has a nature to naturally goes into recipient genome and integrate it themselves. So again, it has to be encapsulated in the viral vector and goes to the nuclei and then goes into the recipient own genome, and then from there to produce RNA and the proteins. It could be permanent. And other, I think I can skip this quickly. So this is example how the DNAs get integrated. Okay, so the second part, so type of treatment and different approaches. Is it for me? Is it for you? So importance for this part of talk is that you need to know the variant. The variant is actually the mutation. So I'm using the term variant here for the mutation. So variant meaning that the error of your DNA or different component of your DNA. So I call variant for that. Different genetic mutations, types, missense versus nonsense mutation in PFIC requires distinct therapeutic approaches. So this is the message on this section. So what is your genotype? This is the one thing I want you to take home today. you want to know your genotype. So genotype is the type of variant in your DNA sequence. And this is the example when we ordered genetic test clinically, and a couple of weeks later, we got the report. And this is totally different disease, but I want to use this as example. So you get this name of gene. This is the gene for alpha-1 antitrypsin. But if it's a PFIC, you get the ABCB11 for BSEP. So things like that. And then you get this, you can ignore this part. And then you have this two component. One is the actual DNA position. So the type of DNA nucleotide becoming A type. So G type of nucleotide, the code of DNA becoming A. And this is the protein type of it. So those two are saying the same thing, but it's just a different expression here, description here. So this means the your protein type of amino acid, E version of amino acid becoming K version of amino acid. And then there is a comment about it. They said homozygous means one from the mom, one from the dad, both has this mutation. And then they comment about this is pathogenic, means this is disease causing. And there is another example for ABCB4, and these are also, this is the missense mutation version of it, and you get report like this. So you have the exact same report. This means you have a homozygotic mutation. So missense mutation is the mutation that change an amino acid in a protein. So it's just a change of amino acid. And the nonsense variant is the mutation that introduces stopping code prematurely, meaning that when the DNA become messenger RNA and the messenger become protein, the synthesis of protein stops in the middle. So this causes the truncating protein. So start with the missense mutation, missense variant, missense mutation that changed the amino acid in the protein. So this is the DNA code, and then each 3 DNA code become 1 amino acid. And if you have a missense variant, let's say if this G becomes C, the amino acid changes from this amino acid to this amino acid. So the protein, the size and overall picture look similar, but the one point of this amino acid is different. And sometimes when the amino acid changes, the entire protein shape changes. So even though it's only one amino acid changes, sometimes it's okay, sometimes make a huge difference. So the problem of this missense variant is reducing protein/gene amount and reducing protein function. For the BSEP example, less bile acid pumping, change the location inside of cell. For example, BSEP cannot go to the surface of the cell, cannot pump. Okay, so going to the nonsense variant, this is a truncating protein. So this is the code, and normal amino acid chain is like this, but if you had the error here, suddenly you got the stop coding, and then you have to stop making the chain. So you have a very short version of protein. Then oftentimes this component get degraded, so you will oftentimes you miss entire protein from this nonsense variant. Okay, so this problem from the nonsense variant is basically you don't have a protein that you need. So going back to the type of treatments. So missense variant, the best approach is to support the protein, either using the small molecules or some nutrient, like phenylbutyrate can do that too. And also they have a benefit from those therapy, but those are the first step we should try. So example, if you have a missense mutation and cannot go to the location, you can just help those to go to the right place and the right function. For nonsense variant, you don't have a protein, so you have to make a protein. So that's why you need those messenger RNA therapy or gene therapies. Those are most feasible, suitable treatment options. So this is a quick table. Missense variant, you have a less protein. You need to support those protein, and small molecules and nutrients are helpful. And those oftentimes, I would say, treating older or later is probably feasible. In the nonsense mutation, you don't have a protein, so you have to make a protein. And to do that, messenger RNA or gene therapy is feasible. And then those are usually severe disease. So you probably need to start treatment younger. Okay, going to the risk part of new therapies. So I'm just using the gene therapy as an example because there was a lot of interest from the family, and we have a roundtable coming up to talk about the risk. So when I was preparing this talk, I asked the family to give me two inputs. So one is, can you define the gene therapy in your own words? That was the question to the PFIC community. There was an answer for that, means correct mutations, remove bad genes and put the good ones, reprogramming, or several ways to do. So there was a comment about it. What does gene therapy mean to you? The answer was big hope. Already good example for other disease, permanent cure, solution versus others treating symptoms. It comes with the risks depending on types. So that was the input. Okay, so let me talk about the benefit first. So it's directly addressing the deficiency. So it can be cured, and it's oftentimes has a very immediate effect. Can be permanent with one injection. So those are benefits, but I have to talk about this. So the ultimate risk of this new treatment and gene therapy is the death. So there is a report in the history and the recent clinical trial too. So there was a death of clinical trial participant for gene therapy. I have to talk about this. This young man, Jesse Gelsinger, he's probably the first person publicly identified as a death of the clinical trial for gene therapy. It happened 2099. He had this liver disease, OTC deficiency. It's a X-linked disease for the liver, has a lot of ammonia, but he had actually a very mild version because of the mosaic condition. He had just that actually doing well with the restriction of diet and medications. He signed up for gene therapy in Philadelphia, and he immediately had a very severe liver failure. He died 4 days after, at the age of 18, after gene therapy. It was reported as an unusual immune response to the viral vectors. It was an immune storm and had the acute liver failure. Before the death of this clinical trial, there was actually 2 patients had the very serious side effect, which were not reported. And there was actually death of monkeys giving the similar treatment, but it was not in the informed consent for this person. So those are the big problem. So actually, this is the gene therapy history. So it started around 1988, and there was a very, and then this is the time course, and then this is the interest and also the movement in the field. So people had this idea, everybody excited about it, but there was a lot of technical issues. So a lot of people said, oh, that's not possible. And then technology improved, and adenovirus vectors are coming up, and the people start doing experiment and having excitement, but because of his death, it's a lot things just shut down. And after that, people improved, changed the technology, and then another wave of interest coming up, and then we had another clinical trial, but because it was a very different condition, I have to say, but there was a cancer after leukemia happened after gene therapy. So the people's risk became more severe, and then now it's more technology coming out. I think we're in this phase of increasing interest in technology with improved safety. So we are on this wave of interest and safety, and I think we are on upsurge for the field interest. Other potential risks for gene therapy is off-target effect. Cancer is possible. This is something we want to talk about in the roundtable. The gene can get into the DNA, and if it goes to a very critical component of DNA, it can turn on the cancer genes. So that's how the cancer can happen. And also, you are targeting liver, but the virus can go all the body. So it can have unknown or unexpected effect to the other organs, like thyroid or adrenal gland or unknown organs. Okay. Gain of immunity. This is something in the Duchenne muscular dystrophy world. They had this clinical trial and then FDA approval for gene therapy, but they are start seeing immune response. It's kind of natural, logical thinking, because when we do a transplant for BCEP deficient patient, we see the antibody against the BCEP. So if we introduce those genes to people who don't have BCEP, is it like just like a virus vaccine? Is it like a virus effect? Sorry, vaccine effect that the body recognizes that as a new protein. So this is the concern, and it's not just a theory. I think we are start seeing this. Okay, so quickly, trying to map out the risk and with the effect. So if this is the risk, high risk on this, and short-term effect to the permanent effect, this is my total makeup, but it lines up like nice correlation. So if you have short-term, usually it has a low risk because you just get out the body. But if you have a very permanent effect, yes, it's a good thing, but sometimes it can have a higher risk because it's staged in the body for forever. And the other thing is this. So the cost of drug development is like astronomical. This is not $460. It's $460 million to make one drug. So this is also, I was making up, but I thought it's interesting and I need to know the safer drug versus high-risk drug, and the funding goes like this. So the more funding and the investment means speed of development. So if it's safe, they get more funding and then they will get more developed faster. But if it's not so safe, they get less funding and takes time to develop. And this nutrient, they cannot get patented so well, so they not get so much funding. So it's safe, but it's not get funded so well. So the summary, so for the family, prepare the future treatment options, know your genotypes, and risks are relative to benefit. And I have to emphasize this more because we just talked about transplant. So the relative to what? Because we have a very, very effective treatment, we just got from Dr. Akihiro about transplant. Transplant is very successful and a very effective treatment. So in the future, when the new treatment, new therapy comes up, we have to compare to this very effective treatment of transplant versus things coming up. Okay, so acknowledgement. Thank you very much for the support, collaborators, mentors, consultant. And I want to specify the funding from this Peter and Tony College Research Award. Those are the support background for Carl Center. And special thanks for Richard Thompson, Benjamin Snyder, Jim Squire, and especially Jim Hybe. He is the one navigated me to be in this field and families and patients. And this is the last slide for connecting to the first slide. Thank you very much for listening.
Well, thank you, Aki, for a really nice overview of all of molecular biology in 20 minutes. That's incredible. Good job. Are there any questions for Aki? or has he explained it so well that there are no questions?
Okay, so thank you very much for breaking that down. I think I understand.
Okay.
When I asked in the research workshop I asked Dr. Grumpy about, does this mean there is reversibility? And he gave me an answer. What I understand is that it's a way to, you're working on mitigating risk in that specific treatment, is what I gathered from that.
Yes.
In a very risky therapy.
Gene therapy you're talking
In a
about.
very risky gene therapy. So that the treatment's still up there on the high risk, but you're working on mitigating the risk through other avenues. And so when the treatment becomes available or in trials, you have safe ways to approach this in patients. But the treatment is still risky, or it will still be risky.
Yes.
There's balance. I just want to make sure I understand, because I think we're going to talk about risk assessment.
Yes. Treatment, so gene therapy, because we use virus and because of those mechanisms, it is risky. But we, day to day, everyone trying to improve the safety, reducing the risk. Yes. Because we believe this is very effective treatment if we can get the safety cleared.
Thank you.
Okay.
I just want to make a comment. Jesse Gelsinger was a long time ago, and it was a particular kind of gene therapy vector adenovirus, which really is not being used or considered for use anymore. So I think from a risk perspective, AAV gene therapy is quite safe. I mean, at this point, hundreds of patients, including very young children. The biggest success story is a neurological disease called spinal muscular atrophy, which is a condition that used to be lethal before the age of 1, typically. And those kids are now getting AAV gene therapy and are walking and in good health at age 4 or 5. There have been a couple of side effects in that group specifically, but given it's always a risk-benefit thing, just like with a liver transplant. I would say the risk from liver-directed gene therapy with AAV is at this point very low. I mean, that's my personal assessment. We can discuss it some more at the roundtable, but it is quite safe. The questions I have more for that particular kind of therapy is the durability, and is it really a one-time injection and a cure? That's where I have my doubts, and I think we might need different strategies. But from a safety perspective, I would have no problems recommending to any of my patients to go for it.
Thank you.
Yeah. And not only an adenovirus vector, but the mRNA technology has really improved dramatically. And how many millions of people have gotten the COVID vaccination with no adverse effects? Sure, there are a few adverse effects, but we're talking somewhere in the range of 1 in 10,000. And in fact, there's Arcturus is now doing a trial of mRNA delivery of OTC enzymes. So,
Yeah, I got the adverse event from the placebo. Yes, please.
All right, yes. First thing, of course, technology is on the rise, and a lot of treatments and developments has been moving, and we never want to doubt that, because you have to do the tests and see everything to continuously get better. But for many of the families and other people that are following, I may have to talk to you afterwards, maybe not. You let me know. What is the best way to follow in real time in the developments of what's going on? And the reason why I ask that question is it's very similar to even real estate. We can understand that the market, you have to follow it for some time to understand the best moments and times to wait, to fold, to pay, to do this. We know where we are. what they need to do next, and then you get more information. And then that's also how we get more people involved, which could also help the rise of these treatments and moving it more and on. And then whether if it's some certain risks or not, you can get more parents and families involved. So that's why it brings it back to the same question. What is the best places where we can be to follow and to understand and be on the same page as you real time in this developments?
So this conference is the best one. So every year,
Yes.
this year, this conference, we will update you.
Okay.
Yeah.
Anything in between
Um,
that I can read to be prepared beforehand.
I think
that
it's
I can follow
at this
you.
point of this, the top part of that therapy, once a year is fine. But the lower part, probably the once in 6 months, we will probably have update.
Okay.
Yeah.
Okay.
It's moving. It's moving.
Awesome. That's all I have.
Yes.
Great presentation, Aki. I just want to introduce maybe a different way of looking at risk besides low and high, and that is unknown. And I actually think with gene therapy for a child gene therapy directed to the liver for a child with underlying liver disease and may even have fibrosis and may even have cirrhosis, that we don't know the risk. For instance, the SMA therapy that Marcus just mentioned, which is, by the way, not given systemically, but given into the spinal fluid, SMA therapy is associated occasionally in children with no underlying liver disease with acute liver failure. And they've had to be rescued with high doses of steroids. Luckily, all those children have survived and then had good outcomes from the therapy for what it was intended. But those children had no underlying liver disease. So I think it's more an unknown risk of gene therapy in a child with significant liver disease. That doesn't mean it shouldn't be pursued on an investigational basis, development basis, industry involvement, et cetera. But I would say we just don't know the risk. And we don't know if it's high risk, low risk, medium risk, and we should just approach it like that. The trials will tell us with data, and that's what we should really base these judgments on once they're in trials. So I just wanted to put that view forward. Sorry, Marcus.
Yeah, and it's true. There's actually there's been another example where there was a myopathy they were trying to treat, and it turns out the protein they were introducing with AAV with a muscle target intention is also expressed in the liver. And when the AAV vector, when given systemically, did go to the liver, it actually caused hepatic failure and death. But it was not the AAV that did it, it was the transgene that ended up being toxic when expressed at high levels in the liver. So it's not zero risk. And with any given new vector, that's why the FDA requires a specific regulatory path for each of these. We just do not know. But there's a second answer to his question. So there's the American Society for Gene and Cell Therapy annual meeting, and it's going to be in 2 weeks in Baltimore. There are, at this point, 8,000 attendees, industry and researchers, and that whole meeting is about gene therapy for all kinds of diseases. So, and that's where I go to get my update. And it's amazing how much the science progresses from year to year. It's in a logarithmic growth phase, not just from the scientific perspective, but it used to be just a bunch of scientists that went there, but now I would say half of the attendees are from industry, and there's a real interest in the pharma industry to bring gene therapies to the clinic for all sorts of diseases. So I don't know what's going to be new in 2 weeks, but I'm sure there will be something that I don't know about yet.
Thank you. Kind of quick comment for Dr. Sokol's comment. So, but we don't know. We don't know the true risk for treatment, but we have to study. So there is one way to do the study is that if someone from this disease going to the transplant, the liver will be taken out. And the liver will be, usually we just fix it and put it in formalin and that's it. But there's a way, just Dr. Willenbach just showed me, that they can put on a pump. They can put on a pump and have a vital organ to circulate blood. So we can have a PFIC liver in the circulation system and have it for a week. Holger, you can have a liver for a week. So that's the opportunity. We can try medication. We can try the therapy for those explanted liver only if patient agree to consent. I'm just throwing out there. Thank you.
Thank you. We'll take a very brief